Welcome to our module on megakaryocytopoiesis. So thrombopoiesis or megakaryocytopoiesis is the process of formation of platelets. Platelets are one of the key blood cells found within the blood, alongside the red cells and the white cells. We'll talk about the different steps in the process of megakaryopoiesis. First, we'll start with an overview, and then we'll dive deep in talking about each of the different parts or steps of thrombopoiesis, as it's usually called. And then we'll talk about some pathologies later on that has to do with the formation of platelets. This is a picture showing us the overview of thrombopoiesis or megakaryocytopoiesis. The process starts with the division and differentiation of stem cells. The multipotent hematopoietic stem cells in this case will differentiate into progenitor cells. Progenitor cells would eventually divide and differentiate into precursor cells. So the precursor cells that we see are what we call the megakaryoblast, and that will mature into the megakaryocyte before platelets are eventually formed. And these platelets would eventually enter into the bloodstream to be part of the blood cells. Let's talk briefly about the processes. Remember that the stages of hemopoiesis includes a stem cell stage, a progenitor stage, and a precursor stage. This is not any different for the thrombopoiesis or this process of megakaryocytopoiesis. It also has its own set of stem cell stages, progenitor stage, and precursor stage. I won't talk about that now. So starting with the stem cell stage of thrombopoiesis, platelets generally are derived from the same set of cells that the starting point for erythropoiesis, which I talked about in the previous module. These cells are called the pluripotent hematopoietic stem cell. These cells will differentiate into two major substem cells. This includes the myeloid stem cell, or what they call the GE-M committed stem cells, and the lymphoid committed stem cell. And our major focus here is on the myeloid committed stem cells, because this is where the entire process of thrombopoiesis will stem from. So the GE-M cells, or the granulocyte, erythroid, monocyte, and megakaryocyte cells, or stem cells, committed stem cells, further differentiate into three major forms of progenitor cells. So they could either differentiate into the burst-forming unit erythroid, and this is what happens when the stem cell goes through the line of erythropoiesis, which is what we talked about in the previous model. Or they could give rise to the burst-forming unit megakaryocyte, which is what occurs when a stem cell wants to go down the path of thrombopoiesis, which we'll be talking about in this module. And then finally, you have the stem cell eventually or likely differentiating into what we call highly proliferative potential colony forming cells. And this is what occurs whenever a stem cell wants to go through the process of forming white cells. Since this model is focused on thrombopoiesis, that means that the stem cells tend to eventually give rise to burst-forming unit megakaryocytes to go through the process of thrombopoiesis. Now let's focus on the burst-forming unit megakaryocytes. These are the first progenitor cells in the process of thrombopoiesis. They are derived from the GE-M stem cells, as I mentioned. And because they are progenitor cells, remember that progenitor cells are not morphologically identifiable. They can't undergo self-renewal, and that's contrast to the stem cells which we just talked about, which can undergo self-renewal, cell differentiation, and cell division. For the burst-forming unit megakaryocytes, which are progenitor cells, they can only undergo cell division and differentiation, but not cell renewal. In the case of the burst-forming unit megakaryocytes, although they are not morphologically identifiable, how we tend to identify them is by the presence of some glycoproteins on their cell membrane. So they particularly express the proteins we call glycoprotein 2b and the platelet factor 4 receptors on its membrane, and this is one way we can identify them. The burst-forming unit megakaryocytes would eventually undergo cell division and differentiate to form the next progenitor cell under the lineage of thrombopoiesis, which is what we call colony-forming unit megakaryocytes. The colony-forming unit megakaryocytes are the second progenitor cells under the process of thrombopoiesis. In contrast to the burst-forming unit megakaryocytes, which is where they are derived from, they tend to express more of those proteins I mentioned, the glycoprotein 2b and platelet factor 4, and this is one way we can distinguish them from the burst-forming unit megakaryocytes. And then finally, the colony-forming units megakaryocytes undergo cell division and cell differentiation to eventually become what we call the megakaryoblast. The megakaryoblast, we'll talk about that in the next slide. They are the precursor cells of the lineage of thrombopoiesis. The last stage under the process of thrombopoiesis includes the precursor cell stage, and the first cell in this stage is the megakaryoblast. Megakaryoblast are small sized cells with very large single nucleus. The nucleus tend to be large enough to occupy the entire space and make the cytoplasm to be hardly visible. You can see the picture of the megakaryoblast in the image below. You can see how the nucleus takes a large chunk of almost 98% of the entire cytoplasm. The cytoplasm just barely visible. The megakaryoblast tend to express platelet-derived growth factors on its receptor. This is one way we can identify them. Remember that because they are precursor cells, we can morphologically identify them under the film, and which is what we are seeing on the screen. And megakaryoblast tend to undergo a form of cell division very unique to the process of thrombopoiesis. This form of cell division is called endomitotic cell cycle, and this would lead to the formation of the next set of cells under the precursor cell stage called megakaryocytes. So quickly, before we talk about the megakaryocytes, which are the next precursor cells, it's important to understand what happens whenever megakaryoblast wants to form megakaryocytes. Megakaryoblast tend to undergo that endomitotic cell cycle. And what practically occurs is that just like every other form of mitotic cell cycle, which tends to proceed from prophase to metaphase to anaphase to telophase, and then finally cytokinesis. In the case of a megakaryoblast, the telophase stage tends to not occur. So there's always a failure of telophase stage occurring, and that failure of the telophase stage occurring for mitotic division is what leads to what we call endomitotic cell cycle. Explain what that means in the next slide. So megakaryocytes are the second set of precursor cells. They are gotten directly from the cell divisions that occurs from the megakaryoblast. Remember that megakaryoblast undergoes a form of cell division we call endomitotic cell cycle, and that usually tends to proceed a similar way to other forms of cell division. The difference is the absence of telophase occurring. So what does that mean? So what we notice is that megakaryocytes have a multi-lobed nucleus, contrast to the megakaryoblast that has a single nucleus. And how that multi-lobe nucleus was gotten is through the process of the endomitotic cell cycle. So the nucleus increases in amount with every cell division, and this is due to a process we call endonuclear reduplication. This is practically the same thing I'm talking about in relation to absence of telophase. So imagine a scenario where you have a cell that is about to divide. Let's imagine that this is a megakaryoblast, and normally a megakaryoblast will have a single nucleus, and if a megakaryoblast undergoes normal mitotic division, we have one cell becoming two cells, and those two cells are separate. They have separate nuclei, and they have separate cytoplasms. But in the case of a megakaryoblast, what tends to occur is that instead of having the cells dividing into two separate cells with separate nucleus and cytoplasm, what occurs is that we have a single cell, the megakaryoblast, undergoing cell division, but getting stuck at the anaphase and telophase stage. So telophase stage is started but not completed, and what happens is that we have a single cell being formed. That single cell is going to contain two nucleus connected together. And those two nuclei connected together is what gives it that multi-lobed appearance. So it's still the same megakaryoblast, but the two nuclei that was formed after cell division remains within a single cytoplasm, and that is what we call endonuclear reduplication. Please just note that the endonuclear reduplication is as a result of the endomitotic cell cycle, which is due to failure of telophase occurring. So this tends to occur with time, and at multiple division. So we have a single megakaryoblast undergoing cell division to form a single megakaryocyte with two nuclei, so say 2N. Then that same megakaryocyte would also undergo its own fair share of endonuclear reduplication to form a single cell, a single megakaryocyte with four nucleus. So that like that, you have four nucleus, cell division occurring again, and the nuclear reduplication giving rise to eight nuclei. 8 becomes 16 nuclei, 16 becomes 32 nuclei, 32 becomes 64 nuclei, and that goes on and on to form about 128 nuclei. And this is all occurring in just a single megakaryocyte. So one single cell would have lots and lots of multi-loops of nuclei within it. It could have as many as 180 nucleus found there, just because of the fact that telophase fail to occur during mitotic division. And this is a very interesting development. This is what happens at the end of the day, that we have a megakaryocyte with as much as 32 to about 128 different lobes of nucleus within its cytoplasm. You can look at the picture just down below. This is a picture of a very, very mighty megakaryocyte. This is one reason why we call them megakaryocyte, because of how large they are. You see the different lobes of nuclei. You could practically count the amount of nuclei found within the cytoplasm of this megakaryocyte. It's up to about 16 to about 32 nucleus found here. So, and this is practically the last stage of the precursor phase of thrombopoiesis. What practically happens from here is that the megakaryocyte with about 32 to about 128 lobes of nucleus undergoes cytoplasmic fragmentation, and each of the different fragments form will give rise to the platelets or the mature thrombocytes, which is the end goal of thrombopoiesis. So at the end of the day, we have one megakaryocyte eventually giving rise to about 50 platelets after cytoplasmic fragmentation. The more nucleus or nuclei we have in a megakaryocyte, the more fragments of platelets are formed. So if we have about 32 nuclei, we have fewer platelets being formed after cytoplasmic fragmentation. If we have about 128 nuclei in a single megakaryocyte, we have as many as 50 platelets being formed at the end of the day. So this is a picture showing us the different steps of megakaryocytopoiesis. Again, we start from the hematopoietic stem cells, eventually giving rise to progenitor cells. Progenitor cells would include the burst forming units, megakaryocytes, the colony forming units, megakaryocytes, and eventually that gives rise to precursor cells, which includes megakaryoblast. Megakaryoblast undergoes cytoplasmic endomitotic cell cycle to form megakaryocytes, and this megakaryocyte undergoes further endonuclear reduplication to give rise to a very large set of megakaryocytes with multi-lobed nuclei present within it. And then finally, the last step is usually in the process of cytoplasmic fragmentation, where we have platelets being broken down, where we have the megakaryocyte being broken down into tiny fragments of platelets. So once more, this image gives a better description of the entire process of thrombopoiesis, but with additional input of different factors that tend to contribute to it. You can see the stem cells present at this first step. Stem cells are cells that I said that can undergo self-renewal. Stem cells proliferate to give rise to progenitor cells. Progenitor cells are seen. You can see that they are not morphologically identifiable. Then the progenitor cells undergo cell division and differentiation to form the megakaryoblast. Megakaryoblast would eventually give rise to megakaryocytes, which further undergoes endonuclear reduplication, which is what we can see. So we have 2N, which is two nuclei within a single megakaryoblast, giving rise to four nuclei, which gives rise to 8, which gives rise to 16, which gives rise to about 128. All of these nuclei are found within a single cell, which is the megakaryocyte. And then finally, cytoplasmic fragmentation occurs, and the large megakaryocytes with different or numbers of nuclei within it undergoes fragmentation or shedding to form platelets. The entire process of thrombopoiesis takes about 7 days to occur for the production and maturation of these platelets. So, and this would include, or this will span the process of megakaryoblast becoming megakaryocyte and megakaryocytes undergoing fragmentation to form platelets. So a total of 7 days is the duration of thrombopoiesis. Just like erythropoiesis, thrombopoiesis is also regulated by a number of factors. This includes thrombopoietin, um, granulocyte macrophage colony stimulating factors, stem cell factor, and interleukin 3 have all been noted to be involved in the process of thrombopoiesis. Let's end this segment by talking about the different pathologies that could arise from the process of thrombopoiesis. So you could have an increase in the production of platelets. This will lead to excess platelet within the blood, which is what we call thrombocytosis. You could have a decrease in the production of platelets, which could lead to a process where there's fewer platelets in the blood called thrombocytopenia. We could have abnormalities in the types of platelets being produced, and this would affect their function. This is what we tend to call thrombocytopathy or thromboasthenia. This is an image showing us a scenario where we have numerous amount of platelets more than normal. Usually platelets count should be between 150 to about 400 or 450,000 cells. But whenever we have a large amount of platelets, we call that thrombocytosis, and this is shown in the image here. You can see these smaller fragments or small dots or small fragments just between these are red cells, by the way. If you look at just beside the red cells, you have these small fragments of cells here. These are all platelets. When you have a large amount of platelets and normal, we call that thrombocytosis. Then finally, this is the opposite image or a scenario where we have fewer amount of platelets. You could practically not even count one single platelet in this image. This is due to the fact that they are reduced in amount, and this can be as a result of reduction in the production of platelets. This is what we call thrombocytopenia. And that concludes our module on megakaryocytopoiesis or thrombopoiesis.